Hello everyone, Adesoji doing is my name. It's another time to start the CHM 303, which is Methods of Chemical Analysis. Today we'll be looking into another study session titled Theory of Sampling. We had discussed theory of errors earlier, and this session we would be talking about detailed information on how to sample correctly in order to avoid errors. Now let's take a proper look on theory of sampling. That's our topic for today. We'll be talking about definition of terms, types of sampling methods, sample collection, preservation, and preparation. We'd also be talking about that with respect to solids, liquids, and gases. Now let's look at our learning objectives. At the end of this study session, we're expected to be able to define sampling and its terminologies. You're expected to be able to identify various types of sampling techniques, then discuss sample preservation and preparation, then also describe sampling of solids, liquids, and gases. Now let's talk about definition of terms. We have sampling. It's a means of taking a representative of the population of analysis. Now, somebody will ask me, what is population? It's a large and total area in which research would be done. That is the larger area. Now, sample is it's a process by which it's a process of sampling from a population, and it's a representative of the population. Now, let's talk about laboratory sample. Sampling employed for analysis after processing from the gross sample, which is also called the population. Now, what is gross sample? It's when a sample is obtained without any form of processing, that is raw data. Now, sampling plan, it's a method, it's a plan that ensures the collection of representative sample from the population. Now, let's talk about sampling properly. Sampling, it's a term used to describe the process of selecting a group of items as a representative of others. Sampling methods are actually ways in which samples are carefully analyzed quantitatively, talking about the amount of analytes present, and also qualitatively, talking about the presence of the analyte, that is, to identify the analyte and to obtain our expected results. Sampling is important for a good data analysis in research work, such that a meaningful interpretation of data can be done. If it is wrongly done, then the whole research work is as good as not done. Sampling size, procedure, and response are actually determinant factors of the sample's representativeness. A sample is a portion and a good representative of the entire population. It saves resources, it saves money in terms of time. It also provides a better result that is more accurate and precise than using the whole population of study. Hence, it is much more advisable using a sample size for study rather than using the whole voluminous population in our research work. So, sampling, still talking about sampling, it's actually unrealistic to analyze a very large population. Hence, it's proper that we use a limited sample, that is a collection of limited samples, to represent the entire population, and that's what we call samples. So the process of obtaining our samples is actually sampling. All right. Now, several samples can be obtained and analyzed such that the resulting mean, which is the x bar as in this equation, that is, this is the equation I'm talking about, the mu equals x bar plus or minus ts over divided by a root of n, where our s is the standard deviation, our t is our t-test, and our n is actually the number of the samples, and our mu is the confidence interval. So it's defined as that. Now we have that standard deviation influences the confidence limit. So we also have this formula talking about S naught squared equals to SS squared plus SM squared, where our S naught is the overall standard deviation, and our SS is the standard deviation for sampling, and our SM is the standard deviation for the analytical method. Now let's quickly take a look of this slide. Let's take a look of what we are talking about here. Generally, or we say a generalization is talk about the theoretical population. Now we will streamline it down to our study population. That is, we want to gain access to our population. Then how do we gain access to our population? That is when we now determine a sampling technique. We know how to go about it. Then what about our sample? Which of the sample, what are we interested in? That's when we now determine our sample. So a proper sampling process involves the following stages: defining your population of interest, identifying the sampling method applicable to the research, and state variables to measure. Then you also determine your sample size, you implement your sampling method, and also data collection and review of the sampling process is very important. So take note of those steps. Now let's talk about the sampling of solids. The other time we said we are going to be talking about sampling of solids, sampling of liquids, and sampling of gases. Now we are taking a look at a sampling of solids, and we'll be talking about that in respect to collection, preservation, and preparation. So let's go from collection. How do we collect our solid samples? Obviously, solids are usually a combination of two or more substances or forms, which may range from a larger particulates like iron ores, a solid minerals, a smaller particulates such as precipitates, a solid gels, a sodium hydroxide pellets. We also have a sediment, soils, roofing sheets, iron filings, and animal tissues. So we have so many like that for the larger particulates and for our smaller particulates. So all these ones are obtained with care, especially when they're heterogeneous, in order to have a very good representative of the population of interest. So the sediments and large samples can be collected using a grab sampler equipped with an instrument to scoop up the sediments. So those are some of those things how we can actually collect our samples. Now talking about the sample preservation, these are actually, so many samples can be very prone to degradation in terms of chemical, physical, or biological characteristics. Now, when these are done, they can be composed of volatile or photosensitive substances, and that is why it's actually advisable for us that the samples are stored at low temperature, usually in a refrigerator, so that we keep it in a low at low Kelvin, a degree Celsius, such that the volatility of the solid samples can be reduced by a complete filling of the sample in the container to minimize gaseous spaces. Hence, there is a little or no escape will be observed. Now let's swiftly move to our sample preparation. How do we prepare our solid samples? This initial sample, that is the initial samples of the solids, may be too large or voluminous to analyze, so there will be a need to reduce the particle size. So solid samples require some preparation before a good analysis can be done, due to most analytical equipment requiring samples to be in solution before being quantitatively analyzed. So some analysis, some instrumentation requires them to be in solution, in smaller sizes, so that we can analyze the particular analyte we're interested in. And that's why we need to prepare our samples very well in terms of collection, preservation, and preparation. Now let's go to our modes of sample collection. The other time I talked about the sample collection, it could be in basically three different forms. It could be through your grab sampling, composite sampling, and in situ sampling. And what do I mean by your grab sampling? This is when a single sample is removed from the target population in a particular time and space. As when I mean grab, when somebody grabs something, it gets hold on the person at a particular time. You grab your neighbor, just a one-time catch. So might not be a representative of all of everything, because it's just a one-time. It's a one-chance thing. It's a one-time mode of sampling. So it might not be a representative of your population. So take note of that. Then we also have your composite sampling. It's a series of grab samplings. You have a grab here, a grab there, a grab at the other end. Then there's a togetherness. You add everything together. That is what we mean by composite. So it just gives you one single composite, and it actually saves time, it saves cost, and of course, it increases your volume. Now, let's talk about the in situ type of sampling for solid sample collection. It's such that your analytical sensor is employed to obtain samples by placing it directly in the population of interest in order to allow a continuous monitoring from without the removal of the individual grab samples. Take note, your grab, you take your sample out of your population. Composite, the same thing, but in situ it remains there, just like your thermometer. You put it inside your handy thermometer, you put it inside your sample site. You don't have to take it off your population, and that's what we call the in situ. Here, sampling is done without a physical removal of the sample. So what about our liquids? We have talked about our solids. Let's talk about our liquids in terms of collection, preservation, and preparation. Now, homogeneous solution can easily be obtained using a pre-cleaned glassware or container by decanting or pipetting. So, but for heterogeneous liquids, we can actually shake them properly for homogeneity, and all these ones depends on our mode, our timing, our containers employed. There's some liquid that you don't employ because of organisms. It might react with plastic, and metals too, with another containing metal, maybe stainless steel. If you want to analyze metal like aluminium, you don't use your aluminium steel again or your aluminium container to obtain your sample. So you use something that will not interfere with your analyte, and that is where we are talking about their collection and container. So the container employed is very important. The mode and your timing is very, very important here. So all these ones are factors that determines your sample collection. So the nature of your sample solution determines it, the nature of your analyte determines it, and your intention, what do you want to use it for, it also determines how you collect your liquid samples. Now, talking about the preservation, this actually needs adequate monitoring, and it can due to changes in chemical, physical, or biological properties of samples. It is necessary to preserve these samples for analysis. Samples can be preserved by adequate monitoring the temperature, the pH, the light exposure. Because of these photodegradable samples, there's some samples that when they see light, they degrade, the analyte breaks off. So we have to be very careful about that. So in that case, we might need like a chemical preservative such as formalin. It could simply be added to some biological samples for proper preservations, and some we just not expose them to light. That could also help, like methylene blue dye. When you are dealing with this, you just cover it up. You can use a dark cupboard or something dark so that it will not the radiation will not take off your analyte. So it is important to note the stability of the sample and the efficiency in duration of your preservation method that will be used. Then your sample preparation, most times liquids do not require special means of preparation for analysis, unlike solids. Now let's talk about our gases. Most technical, gases, they're actually most technical than all other forms like solids and liquids. A Teflon bag can be used for collection, or a stainless steel canister can be used as a containing vessel, having filled with a portion of gas pulled into the container using pump. Then cooling obtained in a stainless steel canister changes the gaseous sample to liquid, so we can convert from gas to liquid for easier collection. So solid solvents can also be employed to obtain volatile and semi-volatile gases, while non-volatile gases are usually present as solid particulates, so there is no cause for alarm for that. And all this can be obtained by filtration or glass fiber filters. Then we can also make use of our several absorbents, as sorbents can be used for this sampling. These include like inorganic polymers such as silica gel, alumina, organic polymers like resins of 2,4-diphenyl, phenylene oxide, or polyurethane foam, or for semi-volatile gases. But I want us to take note of this, that there is something we call the breakthrough volume, and this is the volume of the gas that can be sampled before the capacity of the sorbent after it's exceeded. So that is referred to as the breakthrough volume. It's actually measured in your cubic meter per grams of the sorbent. So please take note of that breakthrough volume. That's there here, breakthrough volume. Then let's talk about that sample preservation. There is little or no need to preserve gaseous samples because of their high stability. If there's no escape, no need of preservation. Then your preparation, how do we prepare them? By usually the chemical composition of gaseous samples are homogeneous, so they are stable, so there is actually no cause for alarm. So we have so many types of sampling techniques. We have the probability and we have the non-probability. So probability in terms of, they can call it to be random sampling, and this is when every unit in the population has a chance, usually an equal distribution, which occurs when each item in the population has exactly the same chance. So each of the samples in the population has equal chance, and that is probability. You can say it's likely to be possible for this to be inside. So that is probability, and it can be subdivided into all these: the simple random, the systematic random, the stratified random, the multi-stage sampling, and the multipurpose sampling, also the cluster sampling. All these are actually in details inside our module, so you can check up inside your module for detailed explanation of this probability sampling. Then the other part is the non-probability sampling, and this refers to method of sampling in which some items of population have no chance of selection out of coverage, or where the probability of selection cannot be accurately determined. So it could be of three types. It could have the convenient, which could also be called accidental, anything convenient, comfortable, probability, accidental, anything goes. So we can also have the purposive, and we can have the quota. So usually the non-probability refers to method of sampling which some items of population have no chance of selection at all. So they could be referred to that, and the selection is not in a random manner. It does not allow for estimation of sampling error. So far we have been able to talk about error, the theory of sampling. So, and this study session has also exposed us to different types of sampling techniques, and also implementation of sampling plan with respect to solids, liquids, and gases. Thank you. So I have the self-assessment questions for you. Please just take a look at that and ensure you can discuss with your facilitator, or just write the answer in your notebook. Then also, this is a take-home assignment for you. And on this note, I really want to say thank you for listening.